All right. I know this takes a while, but it seems like I'm moving like a tortoise. But I do think, like I said, you have to go back and you have to think about this and go through it slowly. So now we're going to take a look at the role of ATP in this process that we've been outlining in the last video. Okay. So here's again, we're close up on, we've really zoomed in on the sarcomere, and we're looking at this connection between the myosin and the actin. All right. We said the sliding of actin along myosin to shorten the sarcomere, again, you saw that here, okay, requires the binding, the pull, the detaching, the repositioning, and the repeat. And we call this, right, because it's repeating cycling, cross-bridge cycling. So, right, here's our binding, and the binding can first start, right, when that calcium has bound to the troponin, move the tropomyosin, all right? And in this situation, it's showing that associated with this myosin head is an adenosine diphosphate and some inorganic phosphate. That's what the little I stands for, okay? All right, so, and that's just traditional representation. So the pull. So I say here that myosin, again, has this ADP and, right, so this basically separated ATP, right? So it's a diphosphate, two phosphates, and here's our third tri. So we need to reform this molecule, all right? But it's here, it's associated, okay? So myosin has bound and attaches, right, to actin. These two molecules are released, and you see this happening here. And this release pushes the myosin, okay? So they're released from the head, they're bound to it, and the myosin is pushed. Now it's moving towards the M line, the middle, and we refer to this as a power stroke, okay? Myosin is now considered low energy, right, because there's no associated molecules with it. So to detach it, we add ATP. So it comes off, right? ATP binds, and this pulls the head off, all right? So releases from the actin. If ATP is not available, the myosin does not detach, and of course this is what happens after death, post-mortem rigor mortis, why you stay in a contractile state. Yeah, go figure. All right? All right, so there's an enzyme, ATPase, right, in this area, and this converts the ATP to ADP, and so it comes apart again, all right? But in coming apart from here to here, we get this change, right? Because energy brings about change. And so this is our energy, and now we are changing the myosin, and we say we are recocking it. It's back into a conformation or a structure that it can bind again, all right? So it comes off, it reforms or recocks, all right? We break down the ATP, and now it can attach at a different site along the actin. So, right, it was attached here, it pulled here. Now we're going to attach down here and pull some more, moving it this way. All right, so now we're ready to attach again. And I say as long as there's acetylcholine, calcium to bind to troponin, and ATP around, the cycle, this, right, bind, pull, detach, reform, bind, will happen over and over again. But again, to the limit of the Z lines. I said, think of this like rowing, right? I always think of it not as sliding, but ratcheting or rowing. The paddles are the myosin. They pull, they're lifted from the water for the detach. They're repositioned, recocked, and immersed to pull again. So it is a good analogy to use here. All right? So I say here again, think about what you can achieve with all this action going on. So if we look again, we go out to the overall sarcomere, which is still pretty darn zoomed in, right? And look at it in this cartoon, right? Here we are relaxed, and we've got our myosin with their heads, and we're zooming in on a connection, a possible connection here. And here it's starting to contract, and here it's contracting more. And again, and pretty soon we're going to hit full contraction. We won't be able to move it anymore. So contraction again, actin and myosin binding, that's we call that cross-bridge formation. And if they slide, right, they do that pulling, then we get shortening of the sarcomere, all right? So the degree of contraction, which is due to the amount of force we can generate, right? Which is, again, force is what we use that ATP for. And therefore, the shortening, so the degree of how much we contract and how far we shorten, depends on how many myofibrils are involved and how far the myofilaments can slide. So there's limitations. Now, look, you really have experienced this. So I know it sounds complicated, but think about it yourself. So the force generated, what do we mean by that? Well, if I'm going to move a pencil, I have got to just generate enough force in my fingers and hand and potentially arm, right, to pick that up. So what is the weight of this pencil? It's not very much. So the force I have to generate doesn't take a lot of myofilaments and myofibrils. All right? But if I was to pick up the desk or the chair, I would need more force, right? Because I always have to move things, and this includes you standing upright. You have a certain mass, and gravity has an effect on that mass, your weight. And so to stand upright, you've got to generate the force to overcome that force that's keeping you down, right? So you just kind of take this for granted, but this is what happens every time these muscles work, right? And so we do need to note that you're going to have to vary this force based on what you're moving, and that there are limits to how much force you can get. You ever been in that position where you try to move something and you just can't pick it up? Well, look, you're trying as hard as you can, but you've reached your limit, right? You just don't have enough muscle fibers, enough cells and myofilaments, and therefore enough sarcomeres made up of these actinomycin. You don't have enough to do it. Now, if you lifted weights, right, you did some weight training, you might. Why? Because when you weight train, you add more actin myosin. You add more myofilaments. You add more sarcomeres. And as I add these, I can get more force. I do remind you that the number of muscle fibers, so how much muscle mass you have, is genetic, right? But strength is dependent on the number of myofibrils, as I just said, and sarcomeres, and that can change. Your muscle can adapt. It's still limited, right, to how many, because you still overall have a certain number. You can't produce more muscle cells after a point. But you can produce more muscle mass through more myofilaments and myofibrils. All right? What is the energy source for contraction? Well, we said ATP. We've been talking about this ATP. So this, again, adenosine triphosphate, it's a fairly simple molecule, but it's the only molecule that not just you, your dog, your cat, your lizard, most of your bacteria in your body, it's the only molecule they use, right? So we say it's our energy currency. It's the usable form of energy, all right? Well, what other forms of energy are there? Well, there's, mechanical energy and electrochemical energy and chemical energy and solar energy and yada yada, okay? But also for you, let's just look at the chemicals. We're really talking about a chemical and electrical things in the body. So ATP is a chemical form of energy, all right? You take in chemical forms of energy. How do you know this? Because you measure calories, right? When we have a calorie associated with a molecule, it means that it has potential energy, which means you have to have the ability to take it apart and transfer its potential energy to this molecule ATP. All right? That's what calorie means. So ultimately, carbohydrates, proteins, lipids have calories and therefore have chemical energy. But that energy cannot be used in that form. You have to have the ability to convert that chemical molecular form to ATP. All right? ATP is made predominantly in the mitochondria of a cell. And so therefore a cell that has more mitochondria would have more of an ability to generate ATP, right? It is made most effectively in a process called aerobic respiration, right? And the term respiration means exchange, right? Aerobic means with oxygen. So the exchange that's occurring here is between two gaseous molecules, right? Oxygen and carbon dioxide. The important outcome is this ATP. All right, so don't let the name be misleading. We also refer to this as cellular respiration because it's occurring on the cellular level. Okay? Muscle cells, right, And it said any cell with high energy demands, high needs for ATP, will have lots of mitochondria and will constantly be making ATP, constantly doing aerobic respiration. I do caveat, ATP storage is very low. It's such a volatile molecule, unstable, that you can't store up and make a whole bunch of ATP, even though you're not using it. Think about being at rest, not active. You can't do that. So as it's broken down by this ATPase at that, the myosin-actin interjunction, it has to be regenerated. So we have this mitochondria producing ATP all the time. And how do we keep up with the demand? Well, we have to deliver, the chemical forms like glucose or fatty acids to the muscle cells. We have to deliver other molecules to the muscle cells so that they can keep on this process of conversion of one energy form to another. All right? So I say there are three mechanisms that help us deal with this energy demand. All right? So one of them, and these are specific, some of these are general for the body and some are specific for muscle, because muscle is such an energy needy or ATP needy cell. So in muscle there's a special molecule, creatine phosphate. And creatine phosphate, just shown as a cartoon here, is a storage form. So unlike the ATP, when you have a lot of potential energy around, again, you've eaten well and you're not very active, your muscle cells can make creatine phosphate. All right? And creatine phosphate can be used. It can be converted back to ATP. So not used directly, but it is an easy conversion done by the cell. So I say it can store and release it quickly, but you really only have the capacity or ability within a muscle cell to store a limited amount. All right? But it's going to allow me to keep that contraction going. And 15 seconds may be the difference between life and death in a very quick universe, right? So when muscle is at rest, here's our scenario, resting muscle, energy and ATP would be diverted into making creatine phosphate, all right? And as it's creatine phosphate's made, the ATP is broken down to ADP and PI, but they don't show it on this cartoon. As soon as muscle becomes active, then that creatine phosphate is going to return, through the help of an enzyme, return the phosphate back to the ADP, and you instant ATP, which is really cool. Again, but not for very long, all right? But that's one way we're going to help this muscle keep on working, all right? So another way is that we can do muscle cells, and indeed other cells, can do glycolysis or fermentation. All right, so glycolysis or fermentation. So lysis, the breakdown of glucose, basically. All right, but we use this because these are anaerobic processes in the body. Okay, well, why? Why is this important? Well, to do this, cellular respiration we talk about, aerobic respiration, we said we need oxygen. Oxygen has to participate in the reaction to maximize the amount of ATP. But oxygen can be limited, because oxygen only comes from your atmosphere. It's delivered to the body through the lungs. It has to bind to hemoglobin in the lungs and then be carried by pumped by the heart to the muscle. It has to diffuse from the blood off the hemoglobin into the muscle, and then it has to be available to the mitochondria of the muscle cells. So, sometimes you run short or lack oxygen to a muscle cell for this reason. All right, so muscle cells can carry on anaerobic, so no oxygen. So again, starts with glucose, This is the potential energy, and it produces ATP. It is not as quick as creatine, so creatine would be your first line or first response to a limited, an ATP limited situation. You suddenly become active and you need energy immediately, all right? Once you exhaust your creatine and if you didn't have oxygen, you would do glycolysis, right? The sugar is also delivered through the blood, but it turns out that sugar can be stored in the muscle as glycogen. So it's available on a regular basis for the muscle's needs. The problem is it only produces very few ATP, and the chemical that comes about, pyruvate, becomes lactic acid, and lactic acid can contribute to increased fatigue. All right? We will pick up on this concept on the next And I know you guys are all fatigued right now.